Hi everyone. So today I'll be reviewing the article that used ancestral sequence reconstruction, and the paper also demonstrate that ASR enabled the identification and synthesis of an enzyme form of CRISPR-Cas12 enzyme with a highly versatile functionality. So CRISPR-Cas systems are antiviral defense mechanism of bacteria in which a nucleus of the Cas family cuts a specific target sequence in the viral genetic material. Cas nucleases are directed to the targets by a guide RNA sequence, but they need to recognize the protospace or adjacent motif, and this makes it difficult for these enzymes to be used for any therapeutic target. So this is the problem. Then what's the solution? The fact that CRISPR systems are adaptive, it means that they have undergone a process of molecular adaptation. So scientists wonder if they can rewind the evolutionary history of CRISPR-Cas nucleus and resurrect an ancestral version, and this enshrined form might not have undergone adaptation. So ancestral node selection is a step-by-step process. First of all, to reconstruct the sequence of REGB using ASR, they collected 63 homologs of Cas12a sequences from different species of bacteria. And then second, a Bayesian phylogenetic tree was generated using Markov chain Monte Carlo sampling. The tree was then calibrated into a chronogram, which is a time-calibrated evolutionary tree. And then the internal node represents here the common ancestor of Hydrobacteria, which was selected for resurrection. So they tested Regby's genome editing ability in different human cells like Hector93 and Halo cells, and compared it to Cas12 variant, which has broad PAM recognition. They targeted 15 sites in 4 human genes, and Regby showed strong editing of 30 to 37% at canonical PAM sites here at TDTC and TTTG. It was also able to edit non-canonical PAM sites like TCCT, TCC with 20 to 30% efficiency. And not only this, it was also able to outperform this Cas12a variant at some previously unrecognized PAMs like TCCG or TATT. And this shows that RegB can effectively edit a wide range of targets, and it is much more versatile than the Cas12a variant. Next, we wanted to see if RegB can be used as a molecular diagnostic tool, and for this purpose, a series of experiments were done. The schematic representation here shows the collateral cleavage activity. This plot is the cleavage activity at different temperature. At 37 degrees Celsius, both enzymes show similar activity, but Cas12a was slightly more active. But at room temperature, cleavage activity drops significantly for both enzymes, but RegB retained better functionality. And this shows that RegB is still effective at low temperature. The next two plots here show the sensitivity of RegB and Cas12a at 37 and 25 and 37 degrees Celsius respectively. At 25 degrees Celsius, the RegB detects DNA down to 2 picomolar, but Cas12a has a higher detection limit of 10 nanomolar. This shows that RegB is better suited for detecting low abundance DNA. At 37 degrees Celsius, RegB and Cas12a perform similarly at higher DNA concentration, but RegB still shows better sensitivity at low concentration. And then finally, RegB was applied in bacterial pathogen detection using 16S recombinant DNA. So 4 pathogens were chosen, and RegB could successfully detect all 4 pathogens, while Cas12a failed due to its strict PAM requirement. So why this work was so important? It shows that by using ASR, an ancestral form of Cas12 was found, and this form can target any nucleic acid because it doesn't have any PAM uh, restriction, recognition restrictions, and that's what makes it a multi-purpose tool for gene editing. Thank you so much.